Yes, good morning. Today now we are going to see about this process of transcription in prokaryotes, lower organisms. So what is transcription? DNA is converted into RNA. This is the first step in protein synthesis, and this process of transcription in prokaryotes involves three important steps: initiation, elongation, termination. Just give an outline and we'll see these three steps in detail inside, right? So initiation means nothing but already I said the promoter site is identified by RNA polymerase and which consists of core enzyme and sigma factor. That is called initiation. And in elongation is the second step where the RNA polymerase it move ahead and it form a separation of the two strands, unwind and form a transcription bubble. And the RNA polymerase remain inside. What is the sigma factor is left outside, and what is synthesized here? RNA is synthesized, which is having complementary base sequence to the already existing strand, isn't it? Now, the thymine in DNA, if it is adenine here, it is replaced by uracil instead of thymine in RNA. Next, in the termination step, the terminator region is identified, and this RNA polymerase along with another new factor called Rho factor, it left outside and newly synthesized RNA is coming out. Okay, so from the DNA strand, RNA is formed. So this process called as transcription process. Now we'll see in detail one by one about these three steps. The first one, the process of transcription in prokaryotes. RNA is synthesized from DNA, that is called transcription. So there are three types of RNA: mRNA, messenger RNA, tRNA, transfer RNA, rRNA, ribosomal RNA. So what is the function of mRNA? It acts as template, basic strand. Transfer RNA, which carry the amino acid and it can read the genetic code. That code we'll study in the next class about the genetic codes later. So tRNA, which carry the amino acid and it read the genetic code. rRNA is nothing but ribosomal RNA. So it is very important structural role it plays in protein synthesis. It acts as catalyst, right? So what are the three types of RNA? Messenger RNA, mRNA, transfer RNA, tRNA, ribosomal RNA, rRNA. These three are having different functions. For protein synthesis, usually all the three RNAs are required. So in prokaryotes, only one RNA polymerase it is involved attached to the promoter site. It can transcribe all the three types of RNA. For protein synthesis, all the three RNAs are required. Only one RNA polymerase it can transcribe all the three types of RNA, right? Prokaryotes, only one RNA polymerase, it can perform all the three steps or stages of transcription. What are the three stages of transcription? Initiation, elongation, termination. Right? Now, the single DNA dependent RNA polymerase, it can recognize and bind to the promoter site and it can initiate and starts the transcription process. And this process called as initiation. So what is promoter? The polymerase enzyme binding sites are called as promoters. Apart from this, it uses as a substrate nucleoside triphosphate. It is act as substrate. Okay, can you understand now? There are three types of RNA: mRNA, tRNA, rRNA, and only one RNA polymerase in prokaryote. It can transcribe all the three types of RNA. Now we are going to see about the three stages of transcription one by one. How the DNA dependent RNA polymerase it bind to the promoter site and it initiate the transcription process. So after the initiation of transcription, that is binding of RNA polymerase to the promoter site, it is already started or initiated. This polymerase continues to elongate. So what is first step? Initiation. Second step, elongation. It elongate the RNA. RNA synthesis begins, adding one nucleotide after another to the growing RNA chain. So RNA chain is synthesized, which is complementary to the one strand of DNA. Next, what is the third one we are going to see? Actually, only a short stretch of RNA, a small piece of RNA, it remains bound to the enzyme. It is already bound to the RNA polymerase enzyme, and it reaches the site called terminator site. At the end of this transcription, already existing, newly formed that nascent RNA, it falls off, it come out. And also, the RNA polymerase also left out. Newly formed RNA and RNA polymerase enzyme left out. So what are the three steps involved here? Initiation, elongation, termination. We'll see one by one. So what is the first step? Initiation. So what is initiation? Already we have studied. RNA polymerase only initiate the transcription process. This is the double-stranded DNA. Double-stranded DNA in only one strand. You can see 5' to 3' and only one strand. It consists of promoter site. Yellow color is called promoter sites. RNA polymerase, it can recognize this promoter site by data box and it can attach. So here the RNA polymerase is associated with another factor called sigma factor and binds to the promoter site of the DNA. This is a promoter site, it will bind, right? And initiation of transcription takes place. Now coming to the point, RNA polymerase along with the sigma factor, when it attaches to the promoter site, what is initiated? Initiation of transcription. Can you understand the first point? So transcription occur in only one strand of DNA. RNA synthesis for only one strand of DNA will occur. So what is the second step? Elongation. Formation of RNA started. Now in elongation, once initiated, RNA polymerase, it moves upon the DNA causing the elongation of RNA. What is the elongation of RNA? Addition of new complementary nucleotides for RNA. But if DNA is having adenine, for RNA synthesis, instead of thymine, uracil is formed. So like this, complementary nucleotides are formed in RNA. Now for this, what is used as substrate? Nucleotide triphosphate. Now come see here, this from this promoter site, this is the transcription bubble. Both the strands of DNA gets elongated and sigma factor which is present inside is left out. Still, this RNA polymerase, this color orange color, it's left inside the transcription bubble. And here, what is formed here? It is called RNA, nascent RNA. It is started to form and elongate, and the complementary sequence of this RNA is complementary to the DNA base sequence. If DNA is having guanine, RNA is having cytosine. DNA is having adenine, and here it's having uracil. And here thymine is there in DNA, adenine is there in RNA. Like this, a small fragment or part of the RNA is synthesized. So this step, the formation and elongation of RNA by formation of this, what is it called? Transcription bubble, leaving the sigma factor, but what is present inside? This is the transcription, that is the RNA polymerase is present inside the transcription bubble. And this process called as elongation. So what is the third step? Termination. Now coming to the termination of the transcription. So first here promoter site initiation, here elongation. What is the final stage? Termination stage. And this site is called as green color is called terminator site. And RNA polymerase first in elongation also it was present in cell. Now this RNA polymerase consists of another factor called Rho factor. It's a protein. Rho factor is present inside the RNA polymerase. So when this RNA, along with the RNA polymerase and Rho factor, which comes to the end of the terminator site, then this RNA, nascent RNA, mRNA, it falls off. And polymerase, along with the Rho factor, it moves away. Here, see here, when the terminator site has reached that particular mRNA, RNA polymerase associated with the Rho factor, it terminates the transcription and mRNA falls off and polymerase moves away, right? So RNA polymerase, Rho factor, when they reach the terminator site, this is called terminator site. RNA polymerase, Rho factor, they reach the terminator site, there is a termination of mRNA and mRNA falls off. This is the basic transcription which occur in prokaryotes. In prokaryotes, mRNA does not require any processing to become active. So transcription and translation, it occurs simultaneously. அடுத்தடுத்து கண்டினியூஸா டிரான்ஸ்கிரிப்ஷன் என்னப்பா, DNA is converted into RNA. What is translation? RNA is once again converted into protein. Both occur simultaneously, continuously. Many times the translation starts even before mRNA is fully transcribed. Even the translation will be started if the transcription is not yet over. Right, like this continuously, both the transcription will occur and translation will occur. This is because the genetic material in prokaryote is not separated from other organelles by the nuclear membrane. Okay, so because prokaryotes, it is only presence of nucleoids, no well-defined nucleus. So that is genetic material, DNA or DNA is not separated from other cell organelles by nuclear membrane, no well-defined nucleus. So transcription and translation both occurs. It is coupled. Coupled means both occur simultaneously, continuously. Coupled activity in bacteria. So clear? This is called the transcription process which occurs in prokaryotes we will see. Next we will see about transcription occur in eukaryotes next one. Yes.